Welcome. My name is Kurt Cotanch. I'm an independent nutritionist, spent many years at Miner Institute and UVM, managing forage labs and performing research projects on rumen function, digestibility of forages, and so forth. Today's presentation, and I want to thank Susan Briette and Amber Macy for inviting me to speak today. It's based on grazing education, rumen dynamics for grazing advisors out in the field. So hopefully I'm going to present a broad overview of basic rumen function, which at times can be complex, but I think a broad look at how the whole system works will help everybody understand when you go to study specifics on the rumen feeds, how the rumen works. So here we go. Dairy nutrition fundamentals with a focus on grazing. All right, general anatomy of the cow. This is of note, this is the right side of the cow. And we can see feed enters through the esophagus, enters the very large rumen, reticular rumen vat, the big white outline there. And it's a very powerful organ, excuse me. If you ever have the opportunity to put your arm in a fistulated cow, I highly recommend it. It's enlightening as to how strong those contractions of moving that ingesta around can be. The large rumen fills up about 40-gallon capacity vat in the middle of the cow. The reticulum and the rumen are sort of one huge organ, and the omasum is a subpouch just below the reticulum. Its function is a lot of water filtration. The abomasum is considered the fourth compartment of the stomach system. It's a true stomach in that it secretes acids to help digest feed particles and so forth. The small intestine comes after the abomasum. Small intestine is where a lot of the absorption of digested nutrients occurs. And then feed travels through the length of the small intestine to the large intestine and exits the cow. Large intestine, there is some microbial degradation there and generation of VFAs, volatile fatty acids, but most of that is lost into the feces. So the rumen holds about 40 gallons, depending on the size of the animal. And it is loaded with rumen microbes. And these microbes there's a plethora of them. I think there's over 400 different species of microbial organisms in the rumen contents. They vary from bacteria, protozoa, fungi. They all have very specific roles in the rumen. We're going to focus on bacteria. And just for numbers' sake, there's, I don't know how many zeros there. It's like 15 zeros there. That degrade the fiber and feed particles and generate the compounds that the cow can use for energy. So this is a picture of a cow out on grass. And a few things to note is, can you see my cursor? I'm not sure. But there's a little V where the rumen fistula would be in that white pocket up by her rib bones. That's where the fistula would be. And oftentimes looking at that can indicate how full the rumen is. Right now, the rumen is not very full because that triangle is kind of sucked in. When she fills up with fresh forage and feed, that pouch will extend and not totally disappear, but at least be a little more distended than what it is now. My coworker over at Miner helped actually put this little video together, and that is just about, it's a little big for that cow, but almost the right size for that cow. And you'll see the rumen extends way far forward and way far backward. It's a huge chamber. That's probably the low point of the amount of ingesta a cow actually holds. You certainly never want them empty, and you don't want them even near half empty. The rumen needs to be filled in order to keep, basically to keep all the organs in place, especially the abomasum. So cow's out on grass. She's going to eat. She's going to fill up nearly to the top of her back. There will literally be no airspace in there. And then she'll go lie down, ruminate, degrade some of that feed, and pass it through the rumen to the small intestine. And that's our job as managers of cattle, is to keep that system filling and emptying, filling and emptying. And the speed, the rate at which we can manage that, depends on the quality and the care that we provide the animal. So for today, we're going to focus on these three, well, actually these two, eating rumination and the rumen function. As I mentioned earlier, the small intestine is the site where the final digestion of compounds occurs, enzymatic degradation of proteins and starches that survive the rumen. The cow absorbs these nutrients in the small intestine. She absorbs her VFAs, or volatile fatty acids, in the rumen. So as I go further on, I'll show you the distinction between rumen and small intestine absorption. But focusing on eating and rumination, um, let's start there. It's a simple concept, but it's the key to the whole functioning and profitability of a cow. As far as time budget goes, eating generally should take 3 to 6 hours. If it takes longer than that, that tells me that the cow is spending too much time, too much energy. There's not enough feed for her to consume within those 3 to 6 hours. She's spending more time eating um, than optimal for a profitable dairy cow. The function of this is to get as much feed in as quickly as possible. It's all about bolus formation, chewing the feed particle and getting it to the rumen as quickly as possible. The primary event that happens at this point is insalivation of the feed and the bolus. She's making a slippery ball in order to swallow it. There's a little bit of particle size reduction, but actually not much, just enough to make that bolus and get it down. Rumination, cud chewing, that rhythmic 60 to 70 chews per bolus, swallow, regurgitation, and repeat. Usually those bouts occur in 60 minutes or longer periods of time. The cow should be spending 9 to 10 hours comfortably ruminating. We could give an entire lecture about rumination, and it's very similar. If you look at brainwave patterns of cattle during rumination, it's very much like REM sleep. So it's a form of rest for the cow. And when they're very comfortable, they will ruminate that 10 hours a day, if not more. And that is ideal, not only for processing food, but I believe for the comfort of the animal, for the well-being of the animal. So what's happening during rumination? Well, this is where particle size really gets reduced. Greater saliva and salivation of the feed particles. And why isn't salivation important? Because saliva is full of minerals and ions and proteins that help coat the particles and allow for efficient microbial attachment to the feed particles. So without saliva, bacteria would not be able to attach to feed particles as well as possible. Particle size reduction is almost a by the way type occurrence. There's a huge, debate of you need to reduce particle size in order to make feed particles exit the rumen. And it really turns out that particle size reduction is synonymous with increased surface area and cell destruction. In other words, we're reducing the buoyancy of feed particles, increasing the specific gravity, increasing the density, so that feed particles finally sink in the rumen rather than floating. And we'll show you a picture of this later, uh, in order to enter the fluid pool and pass to the rumen. If you watch cows chew, you'll see that they're not biting. They really crush and shear in a circular jaw motion that mills the forage and feed particles. It crush and shears for maximal surface exposure to allow microbes to attach and degrade the fiber. And of note is that degradation often of cells, of plant cells, often does occur from inside out. So there's so much protective layer on the outside of fiber particles that the bugs need to get exposure to the inside secondary cell wall to attach and degrade the fiber. Okay, so microbes need nitrogen in order to do their job, and that can come in the form of non-protein nitrogen, NPN, or peptides, true amino acids, true proteins. They pair up with sugars and organic acids, soluble fibers. The carbohydrates, the CHO is the term, the general term for that broad character of compounds. And they put the protein and the carbohydrate together to grow their own cells and produce microbial protein. So they replicate about every 20 minutes under ideal conditions, and obviously can generate a tremendous amount of microbial mass that eventually gets digested by the cow in the abomasum small intestine. So if we have the optimal amount and variety of nitrogen sources to match up with the various carbohydrate sources, we can maximize microbial growth. And we term this the synchrony of nitrogen to carbohydrate ratios for optimal microbial degradation. And we'll see that coming up as well. All right. Look at this picture. I look at this picture quite often. It's just a good reminder of what's going on in the rumen. Feed enters, carbohydrate and protein. It's structural carbohydrates. There's non-structural carbohydrates that get degraded by rumen microbes into the 3 VFAs, acetate, propionate, butyrate. CO2 is produced as a waste product, and hydrogen is also produced, which combines to make methane. These gases get eructated during rumination and expelled, and that's the primary source of methane from ruminants. The VFAs are absorbed by the papillae of the rumen lining, enter the bloodstream, and are transported to the liver, where the liver will take propionate specifically and make blood glucose in a process called gluconeogenesis. So glucose then travels to the mammary gland. Mammary uses it to produce lactose, the milk sugar, and lactose is the actual driver through osmotic regulation of milk volume. So the more lactose, the more fluid in the mammary, the more milk. Interestingly, acetic acid can be used in the mammary as an energy source along with butyrate as well. A little more simple diagram here. Feed carbohydrates and proteins enter the rumen. Microbial fermentation. Hydrogen is the driver of this process. The microbes produce the VFAs at the bottom, acetate, butyrate, propionate, and produce some waste products, H2O, H2, and CO2, which ends up forming methane that gets eructated out. This is provided just as a general makeup of the fractions of carbohydrate in vegetative material, plant carbohydrates. Generally, we break that down into cell wall versus cell contents. The cell contents are relatively highly digestible, smaller carbohydrate fragments, including organic acids, sugars, starches, fructose, fructans, kind of soluble fiber. That's what the NDSF is, neutral detergent soluble fiber. The neutral detergent is the cell wall structure. Hemicellulose, cellulose, and lignin is part of the constructed material to link cellulose and hemicellulose. Lignin is not listed here because it's not a carbohydrate. It's actually a plastic. It's a polyphenol. So again, here's the same diagram broken up a little bit for us. Soluble carbohydrates, organic acids, sugars, starch, very degradable in the rumen, although variable degradability depending on the quality of all of these. The fiber fractions, the NDF, is composed of ADF, which is made up of cellulose and lignin. If we include the hemicellulose, that's when we have the total NDF. Neutral detergent fiber stands for, if I boil this fiber in a pH 7 neutral, all of these three compounds remain, and that's the best estimate of the total cell wall. If we acidify that laundry detergent that we use, I jokingly say that it's a little fancy laundry detergent, we can isolate cellulose and lignin. Now, these fiber fractions are tremendously variable in their digestibility. They can be really slow or they could be really fast. Lignin is undigestible and is associated with the total digestibility, but not the sole determinant of digestibility. So we used to think that lignin was the best indicator, and it turns out we have better indicators of fiber digestibility now. Okay, what do these carbohydrate fractions end up turning into? Sugars oftentimes get fermented to butyrate, a 4-carbon carbohydrate compound. Starch is broken down into propionate, a 3-carbon structure, and fiber is broken down into acetate, a 2-carbon structure, which makes a difference when it comes to utilization by the cow and the liver for blood sugar production, gluconeogenesis. Butyrate can be used for milk fat production and oftentimes gets utilized by the gut lining tissues. Propionate, we said earlier, goes to blood glucose production and drives lactose production in the mammary and hopefully milk volume. Acetate is a primary component of milk fat production. Skipping to the protein side of things. So on the far left, we have feeds that are made up of true proteins being those that are composed of amino acids, and then NPN, non-protein nitrogen, urea, nitrates, ammonia, for instance. They're compounds with nitrogen, but they're not true amino acids. Feed enters the rumen. The microbes have at it. They will take the free NPNs and they will start tearing apart all of those true proteins and changing. They will deaminate, take the nitrogen off amino acids and reutilize that nitrogen and the other fraction of the amino acids for energy. They will reproduce and grow and multiply, and microbial protein passes out the rumen, digested in the small intestine, and becomes the primary source of amino acids that the cow actually digests. You'll see that some of the protein we used to call bypass or escape protein. Anything that does not get broken down by the microbes can pass through the rumen um, relatively unscathed, so to speak, and those amino acids will be directly digested by the cow. Okay, so one thing to note is that there's a tremendous amount of ammonia being produced and utilized by the microbes in the rumen. And sometimes that amount of ammonia is too much. And like the VFAs, it'll be absorbed across the rumen wall and circulate through the liver and around the body and can actually, it's either excreted or uh, recirculated through saliva to re-enter the rumen. It's a way of conserving nitrogen from the animal's perspective, so that you can feed relatively low protein diets because of this function that the animal keeps cycling excess nitrogen that the microbes might need. Something to keep in mind when we talk about precision nutrition. So here are some short terminologies. Rumen RDP is rumen degradable protein, anything that gets broken down by microbes. RUP, rumen undegradable, escapes the rumen and is digested as is by the cow. So what we have here is VFA production in the rumen. That's the primary energy source. And the cow gets most of her proteins in the small intestine. So an interesting thing, we have two different facets of how the energy and protein is provided to the animal. So how much glucose does the liver need to produce for that animal to thrive? Generally, 1 kilogram of milk requires 72 grams of glucose, eventually turned into lactose. So if you have a cow making 30 kgs of milk, that means she's putting about 2,160 grams of glucose into that milk or thereabouts. And if the liver is only generating 3,000, there's only about 800 grams of glucose left for the rest of the body. So what tissues need glucose specifically? The brain and the immune system they can only use glucose. Muscle tissue, fat tissue, other tissues in the animal can use the VFAs, um, acetate and butyrate, for energy. But the brain and immune system require glucose. So if we have an immune event, cow gets sick, uh, injured, just upset any sort of damage or challenge, mastitis, for instance, heat stress, anything that requires the activation of the immune system, there's a tremendous amount of glucose required to run that. And what happens? We're going to steal it from milk making in order to conserve it to run the immune system. Okay, skipping to just a reminder here, in order for digestion, optimal digestion of the sugars and starches and pectins, all of these different microbes require specific NPN, peptide, and amino acid sources, along with minerals and in many cases sulfur. So relative to grazing systems, store feed of baleage, We'll see feeds should have plenty of fiber. Hopefully it's good fiber. And because it's a baleage, it's been fermented. The sugars have been utilized to preserve the baleage. So sugars are very low. Starch isn't going to be there. It's a grass usually in baleage. Pectins could be there. Pectins is the generic term for soluble fiber, and grasses have a tremendous amount of fructans, which get lumped into this soluble fiber portion. So this is proportionally uh, the amount, the, the, the schematic of the carbohydrates in baleage. Compared to pasture, fresh grass, out in the sunshine, she's eating it as it's photosynthesizing. You can have a tremendous amount of carbohydrate coming from sugar, nothing from starch, a fair amount from pectin, and a fair amount from the cellulose, the fiber of the fresh green pasture. So in fresh grass, the schematic of the sugar sources is very different from stored feeds or fermented feeds in particular, which indicates you've got a different need of protein sources, nitrogen sources for the rumen bugs. I like to include this one. This is kind of curious because this shows you how well the amino acid profile of rumen microbes matches up with milk relative to these essential amino acids. And isn't Mother Nature wonderful? Because it's very understandable why milk has its profile when the rumen bugs match up so tightly to providing those amino acids. If we do the same thing with other types of feeds, like canola, soybean meal, gluten meal, we can see that all of these feeds, relative to the proportion of essential amino acid in milk, how well they match that need, they all have weaknesses somewhere. The rumen microbes pretty much meet 100% of the amino acid as it occurs in milk, whereas canola meal, very weak in leucine, isoleucine, lysine, only meets or surpasses any amino acid in arginine and phenylalanine, threonine. Soybean meal, same scenario, provides far more arginine than is required to make milk, but its shortcoming is it lacks methionine. Uh, Corn products are very deficient relative to the requirement for milk production in lysine and so forth. So maximizing rumen microbes is the key. This is a schematic in one of the nutrition programs that shows the synchrony of whether or not you're providing uh, the protein fractions, the NPNs, the amino acids, in the right availability, degradability relative to the degradability of the carbohydrate in the diet. The carbohydrate amount is depicted in the green bars in kilograms fermented, degraded per hour in the rumen, and the red is the amount of available nitrogen at that same time point. On the left, we see that early on, pasture grass, this is a pasture diet on the left, there's extreme excess of available nitrogen relative to the amount of fermentable carbohydrate. So hence, where does this nitrogen go when it's in excess? It gets absorbed and recycled and hopefully available for later on. After the third and fourth hour of consumption, these are eating bouts. There's 4 equivalent eating bouts for the schematic here. After 3 to 4 hours, we see that nitrogen and the fermentable carbohydrate are more in a line on pasture. But initially, there's far too much nitrogen relative to carbohydrate. On the right, we have a conventional corn silage, grass silage, soybean, cornmeal diet, where the synchrony of these diets is better up front. We don't over provide nitrogen relative to carbohydrate. But after 2 to 3, 4 hours, we're short of rumen available nitrogen in this case to match up with the degradability of the carbohydrate. So in this case, the nutritionist would want to go back and say, hey, we've got to improve this diet, providing more longer term rumen available nitrogen relative to the amount of carbohydrate. So it seems complicated, but it really isn't. Once we understand um, the amount of each of the fractions of the carbohydrate, sugar, starch, NDF, and the amount and degradability of the protein or nitrogen sources, we can mix and match and hopefully provide them in the right proportion. So the key is having a good grasp on the degradability of these individual carbohydrate components. That's where it gets complicated. And this is where the forage labs are getting really good at not only analyzing for these specific fractions, but also giving a pretty good estimate of rumen degradability of the carbohydrates. The next key part of this mathematics, so KD is the K for rate of degradability. You're going to see KD of sugar, the rate of degradability of sugar, rate of degradability of starch. The next key factor in the equation of how much gets degraded is how long does stuff stay in the rumen? And that comes down to the rate of passage. How quickly does it move through the rumen? So once you get an understanding of all things do not move at the same speed through the rumen because it's related to degradability. The higher the degradability, the less time it stays there. Slower the degradability, the more time it stays there. Straw stays in a rumen a lot longer than fresh grass. So its KP is much slower than fresh grass. The nutrition programs can only divide this up into liquids and solids passage rates. So they do a pretty good job of showing that two different rates apply. I'm going to leave it at there. Particle size does matter to the extent of surface area exposure to maximize microbial attachment. So you can be too long and sometimes you can be too short because that particle will sink and not stay in the rumen long enough. So this is a picture we took way back working with a farm that was feeding an all grass TMR with a little bit of cornmeal. And this is just water. We submerged the TMR in water. We also collected fecals from the same pen. And this is the idea. You've got to go from left to right within, it might be a day, 30 hours, 24, 30 hours, a little more than that, right? Because they eat the same amount every day, hopefully. Therefore, they have to process, absorb, and pass the same amount every day. But the trick is getting these particles degraded and sinking and flowing in the liquid pool to end up at the bottom in the feces as sediment at the bottom. This is a picture of two forages, quality A and quality B. And what we did was we submerged them, we put them in Dacron bags and submerged them in the rumen for either 0 hours on the far left, 24, the second set, top to bottom, 48 hours, third, and on the far right for 5 days, 120 hours. And what you can see here is that forage A within 24 hours immediately shows that the cells have been opened up. This is only from rumen bacterial. The cows didn't chew this. It's only rumen microbes doing the work here. The cells have been opened up, the pigments have been leached, uh, some tufting, so degradation of those bonding of the cellulose. And as we progress to 48 hours, now we're actually seeing particle loss. I mean, it's being degraded. The mass is less and less. The tufting is greater. By 5 days, it's just kind of a wad of cotton. Forage B had a much lower rumen degradability from lab analyses and from this visual depiction. Even after 24 hours, it doesn't look a whole lot different than what we started from. After 48 hours, we finally see some tufting of those forage fibers. And by sheer mass, we see that even 5 days later, there was quite a bit of material left compared to forage B. And these forages differed by over 5 units of NDFD. Work by Dr. Olba and Allen showed that for every unit of forage fiber NDF digestibility increase, 1 percentage unit increase equates to about an increase of 0.4 pounds of dry matter intake, DMI, which relates to about half a pound of increased milk production and over half a pound when we correct for components. You're wondering why they can actually make more milk than what they can eat? Because you're extracting more energy from that forage material besides just eating more. Corn silage, similar scenario. The greater the NDF digestibility, we get more intake and more fat-corrected milk. So this is a Cumberland Valley data set from a few years ago, and what it shows is that by forage type, legume versus grass versus corn silage versus BMR corn silage versus small grains, we can see that NDFD 30-hour is on the x-axis, and that legumes, in this case, the bell curve, most of the samples were in the mid to low 40s for a 30-hour fiber digestibility, compared to grasses in the dashed green line, which had 50% fiber digestibility, almost a 10-unit increase compared to legumes. Corn silage, about 55% of the NDF is degraded in 30 hours. BMR corn silage, about 65% of the NDF is degraded. So forage variety matters when it comes to fiber digestibility. And oh, by the way, the potential for the best, fastest amount of NDFD 30 is with grasses and small grain silages. Cool season grasses are fiber, cool season grass fiber can be extremely digestible and a very good source for milk production. Maturity is actually the factor that matters most within a variety. Obviously, early vegetative fiber is more digestible than a mature woody stem fiber from forages. And these are generated using real data. So I have an array of pasture grass, vegetative grasses, proceeding from boot to emerge to seeds and stem development as we go to left to right. The orange portion of the bar is indigestible. U stands for undigested at 240 hours as a percent of the total NDF. Same with the yellow is slow, is the portion of fiber that is fairly slow digestion. It takes 30 hours or longer. It's the difference between 30 and 240. The green is the portion of fiber that degrades within the first 30 hours of rumen exposure. And as you can see, in a pasture grass, nearly 70 to 75% of the total NDF being about 40 for pasture grass, 70 to 75% digests very rapidly in the first 30 hours. As we proceed to boot stage of growth and emergence, the structural carbohydrate of the orange and a yellow proportionally increases. And we see that at the boot stage, NDF might be at 50 on a grass, and about 60% of that NDF is going to be degradable in the first 30 hours. 40% is going to be either slowly digested or undigested. And as we get to straw has a little bit of fast fiber. In fact, if you grind it up small enough, you can get a good digestibility value from it. It's not ideal, but it is digestible. But it's mostly woody stem, and about 50% of the NDF is going to be undigested after 10 hours in the rumen. So take-home message here is maturity matters most. Anything below earlier than boot is a good opportunity for highly digestible fiber. Anything after seed head emergence is getting pretty woody. It's going to provide a lot of rumen fill without providing that much energy. This is a graphic from Oregon State, just showing as forage matures, your yield will increase tremendously, but the quality decreases tremendously. So forage yield goes up, maybe 3 tons per acre at full flower, but in this case, your yield of digestible fiber is actually less because it got so woody. Okay, forage analyses. I could spend all day talking about this one. And there's a lot of numbers here. And the reason we generate is so many nutrition programs use different analytes to do their calculations of digestibility. But I'm going to show you the key ones I focus on and that matter most from just the quick look. If you're not using a nutrition balancing program, here's the quick evaluation system that I advise. I mentioned earlier that these forage labs are getting really good at characterizing carbohydrate pools, organic acids, sugars, starch, and the protein fractions. And they're also getting pretty good at indicating the digestibility of those fractions. Oops, sorry. Thing to note is there are 2 versions of analyzing forages. The quick and less expensive is near infrared NIR. Very good means of analyzing forage. The other option is wet chemistry, where they actually will, perform the boiling extraction procedures on fiber and sugars and starches. They use wet chemistry to calibrate NIR. NIR, unfortunately, is not very good on inorganic materials. So if we need precise numbers on minerals, we need to ask for wet chemistry. Okay, I'm going to go quickly through this. It's amazing. You have to remind so many of people of what crude protein really is. It really is a crude number estimating protein. It's simply N times 6.25. And as you can see at the bottom, it doesn't necessarily reflect the amount of true protein. In this case, the estimation of amino acid protein is only 875. The difference between 14 and 875 being 6.5 soluble protein, which they assume is not true protein, but that's not a correct assumption. Uh, Ammonia is a good number to track. Ammonia as crude protein equivalent, CPE. And the column to look for is ammonia as a percentage of the crude protein. How much is CPE? And in this case, 5.4 is relatively low. In some fermented feeds, that number can get over 15%. Generally, you want to keep that ammonia portion less than 12% of your crude protein. The NDF fractions, they're providing all of the data here. Numbers to focus on are NDF and ADF. As a percentage of dry matter over here, 51 and 34. Most labs also provide your NDF on an OM basis, organic matter. So they're correcting for ash. This column does not correct for any ash material. There should not be more than 2 percentage units of ash in your NDF. And in this case, that's right what we're looking at. So this is fine. If there was more than 2% ash between 51 and 49, then we've got a soil contamination issue, which is bad. Bad for rumens and bad for analyses. The litany of 12, 24, 30, and 72-hour digestion numbers here are the NDF digestibility at those hours, and the column to look at is percent of the NDF OM. So how much of the NDF ash corrected is digested in 30 hours? In this case, 60%. That's pretty good. Anything lower than 60% would be a slow digestion process. Anything over 60 is pretty good. And I now recommend 65 or better for high quality forages. Silage acid values are provided as well as sugars. Ethanol soluble sugars are simple sugars, glucoses, for instance, fructoses. Water soluble sugars includes the longer chain fructans, which are highly digestible. It's not a structural carbohydrate, but in grasses, it functions as a moderately structured carbohydrate, but they are water soluble and come out in this fraction. So you'd like to see these above 10%. In fresh grasses, your sugars will be 15% on a sunny day. Your water soluble will be above 15% on a sunny day. If it's fermented feed, then these carbohydrates are utilized in the fermentation and will be in the single digits. This is a complicated issue. Ash, as you can see, is an indicator. Just said the NIR is not great on mineral content, but they're pretty well calibrated. Anything over 8% ash is a possible soil contamination for grasses. If it's over 12% for legumes, it could be soil contamination. And then the VFA profile, again, NIR is okay, but wet chemistry will give you much more accurate numbers on these. Butyric acid is often a culprit, that smelly sock type VFA that can provide limit intake if fed too much and end up in off flavors of milk, for instance. Okay, here's, sorry, so many permutations of evaluating the energy content of forages. And right now, I'm going to focus on net energy lactation, which is the old school one, but more importantly, this ME, metabolizable energy. Anything over 1 is good. If you can get to 1.1, it's great. Anything less than 1 will show you just doesn't have enough energy to do much for the cow. couple of pictures here. We're getting close. This is June wiregrass, a lot of stem, not much leaf. It's a green grass, but it's not providing much energy. This nice juicy orchard grass, a lot of leaf, a lot of juicy stem, high quality feed right there. This dead grass, you can see it's partially green Timothy, but a lot of thatch is left in there. If these grasses are dying, they're giving up energy and don't provide the cow much energy. So here's a few helpful hints on good, bad, and average. Always check the dry matter first. If it's supposed to be dry feed, is it dry? If not, if it's supposed to be a dry feed and it's below 85% dry matter, then that feed is suspect to yeast and mold contamination. Check the NDF level. That gives you an indication of the plant maturity. And again, there's a whole nother uh, talk we could give on evaluating forages for NDF and maturity. But in the mid 50s for grasses is where you want to be at the boot stage. Digestibility at 30-hour, shoot for 65 in that column right here. Your sugars, you want to be in the teens on fresh forages or dry forages as well. Potassium is going to be a critical mineral to watch for dry cow nutrition. High potassium numbers can throw off calcium metabolism. Butyric acid is always a concern, not only for dry cows, but also lactating cows. And again, the energy values, if you're focusing on this sheet alone, these are the two energy values that I put my most focus on, NEL value and metabolizable energy. Now for the decoder ring. So for grass forages in a pasture-based system, early vegetative grasses, your NDFs will be around 40, your 30-hour digestion will be over 70. That results in an NEL value of 0.8, TDN value of 78, and that ME megacal of 1.26. You can make milk with this forage, feed young. They all need that quality of forage to thrive. Late vegetative stage, boot stage, like a baleage, where NDF is around 50. You can expect your 30-hour digestion to be 65, your energy values 0.66, 67. And again, metabolizable energy, megacals is over 1. This is sufficient to meet the energy needs of dry cows, growing heifers, pregnant heifers, and so forth. Once seed heads are out, baleage quality is only average. Your NDF will be 60 or above. Your digestibility will be 55 or so, if that, might be lower. Energy values have dropped significantly to only 0.6, and now you're below 1 megacal per pound of dry matter. This can support maintenance energy values only. The famous heifer hay is not meant for heifers. It's meant for nobody, actually. That NDF is in this high 60s, 70s. Your digestibility is very poor, below 50. Your energy values are very poor. This is going to keep rumens full, but animals will not grow much on this type of heifer hay. Here's a schematic we put up. This is using NRC values of a third lactation small breed cow, medium breed cow, 150 days in milk. And we see by body size, 1,000 pound, 1,200, 1,400 across a range of milk production values. There are predictions on the megacals required to support that level of milk. So if we take a medium-sized Jersey, 1,200 pounds, making 50 pounds of milk, we can see 56 pounds of energy-corrected milk. Megacals required per day are going to be about 46. Your dry matter intake, according to National Academy of Sciences, is going to be 53 pounds. As a percent of body weight, she might be eating 3.6% of her body weight. But more importantly, the megacalories per pound of food required to support 50 pounds of milk is over 1. In fact, this medium-sized 1,200-pound Jersey needs all of her feed, no matter how much milk she's making, to be over 1 megacal per pound of dry matter. That's how we use this chart. So this is a reiteration of what we said earlier. High quality feed provides sufficient energy. It's low rumen fill. In other words, it degrades quickly, provides a lot of energy. It's got fast passage. Late vegetative feed, moderate level of rumen fill, takes a little longer to get degraded, does provide energy. Anything, once the seed heads come out, we're talking, it's getting woody. Takes a long time for the rumen bugs to work on it. Fills up the rumen quickly, has a slow rate of passage, and it will limit the amount the animal can eat just because there's a backlog of undigested fiber in the rumen. Question is, do you have enough high quality feed to feed everybody for their lactations? Inventory management, again, another topic worth another seminar. Do you have enough of the quality to support all of your cows at the production level you require? The picture here, do you know what's in that versus, I'm pretty sure I know what's in this bale. Label the bales and have good inventory control. Sarah Flack's book is outstanding, provides a lot of good information, a lot of guidance, especially relative to inventory management, because these estimates of how much cows or various ruminants will eat or require, you'll see in this case, dry matter intake or dry matter demand is the terminology we're looking at. The dogma, this is where one of my concerns is. The dogma has always been dry matter intake or dry matter demand is a certain percentage of body weight, depending on the animal's metabolic needs. Is she lactating? Is she dry or not? So that percent of body weight typically ranges between 2 and 4, depending on what she's doing, lactating or not, and the quality of that feed. NDF, the dogma has been that animals can eat 1 to 1.2% of their body weight as NDF. But again, we now know that the quality of the NDF really matters and can dictate how much the cows eat. So for instance, 1,200-pound lactating Jersey, eating 4% of her body weight means she requires 48 pounds of dry matter. So for a 300-day lactation, she's going to need over 7 tons of dry matter. And you'll have to do the math on your hay inventory and your bales and dry matters to back calculate if you have enough to support those 300 days. If we do the same math with NDF at 1.2% of body weight, it says it only requires 14.4 pounds of NDF. That's what she's going to eat. But that 14.4 pounds is 30% of that 48. So that's saying that same feed would have to be 30% NDF to be consumed to provide enough dry matter. And we know that not even not all pasture grasses, that's a lush pasture grass if it's 30% NDF. In other words, this is causing me concern because they're not in alignment as to best estimating how much the cow is going to eat or how much inventory I need. And what this says is that depending on forage quality, if NDFD is over 65%, 30-hour digestion is 65% or greater, NDF intake can range a lot higher and therefore require me to have a lot more inventory. So we've documented high quality forages of 1.6 to 1.8% of body weight as consumption of NDF. All right, I know that was quite a bit, and every one of those slides could have gone a lot deeper, but appreciate your time. Hopefully this was helpful. And from this family of Jersey cows, I want to thank you for your attention.